it was about 6 months ago that I spoke at one of your previous sessions. And since then, we've filed 10 patents. We've sold 11 instruments for $2 million to one of the largest pharmaceutical research companies, and to a firm which will be sending it up into the International Space Station. And we also raised several $1 million, and we're oversubscribed on our $5 million round. But we actually have opened it up, and somebody who I met here speaking, Sal Buscemi, is going to be leading a round for HRN along with Albert UN. So I started Thrive because I found this incredibly neglected but important area of science that was holding back the whole field. And it's cell culture and cell-based experiments, which is really the basis of almost all biomedical research. I've started 17 companies, 7 have been unicorns, 3 were AI companies, most of the remainder were life science companies. Okay, is this okay? Just hold it. Sure. So started 17 companies, 3 were in AI. One in the life sciences area was very similar to Thrive Bioscience, my current company. And that was Cytic, where we automated cytology labs. Cytology labs read pap smears, the world's most widely given primary screening test. And we totally revolutionized, changed the face of cancer, cervical cancer. 90% of all labs use our instruments. And we're going to do the same with cell culture. It's a very similar but larger market that is really lacking in tools. So next slide, or actually first slide, Marty. Sure. So what are we doing? So we're modernizing with data and automation, cell culture and cell-based experiments. And the title of this talk is Uber knows more about us than researchers know about their cells, which is very sad that a phone app knows more than we do. And the theme is that more and better data will speed successful drug discovery. And that's the real problem that we're addressing, is speeding to market. How are we doing this? With industrial automation. There are no discoveries that are needed. A lot of very clever innovation being brought to an area which simply didn't have before. And we're trying to turn the odds of drug development success so that it's better than the odds at Las Vegas, which literally are better than current drug discovery. Next slide, please, Marty. Sure. So we're industrializing biology, and almost exactly 20 years ago was the first cell line manufacturer, which was at Tuskegee Institute. And this is a picture of it. And you could characterize the situation back then as manual processes, poor environmental controls, poor image and data capture, limited documentation, limited analytics, and non-scalable. Unfortunately, we're the same place today. As one of our customers said, cell culture may be the only remaining island of outdated manual processes surrounded by an ocean of automation. The rest of life sciences has had the benefit of analytics and data and automation, but we still have manual processes and very little data. And that's what we're solving. We're bringing automation, environmental control, huge amounts of data capture that's referenceable and accessible. And we're bringing analytics so that it is scalable and more accurate. Next slide, please. So we have an integrated family of products that do not require FDA approval, which is important. We don't have one of those binary decisions. I ran several pharmaceutical companies, and those are some of the worst moments for management and for investors. There's a tremendous amount of risk in that situation, and thank goodness we don't have that. We sell picks and shovels to all the people who are developing drugs. We have 18 user sites. 60% of those are in industry. And the payback on our instruments is about 24 months. We have some really important collaborations, one with one of the largest Japanese pharmaceutical companies, but also with really key research organizations such as Harvard Stem Cell Institute, Wake Forest Institute for Regenerative Medicine, Center for Genomics Medicine, and Mass General Hospital. We project sales of $4 million in the next year. We just booked a couple million of that. And in 6 years to be at about 250 million. And it's possible to do this because of the vast size of this market. It's relevant to almost all parts of life sciences. There is minimal competition. We've developed an incredible portfolio of 86 patent applications, and we don't have FDA approval, which allows us to grow much more quickly. And we're not dependent on discoveries in biotech or expression of proteins. So we've raised $31 million to date. The vast majority is in fact from family offices, the largest being the Estee Lauder family, which have invested in 2 of my prior companies, both multi-billion-dollar exits. Also the Milstein family, New York real estate family, KP Chow family in Hong Kong, one of the richest families in Hong Kong and Macau. So we're raising the last $3 million of a $9 million raise on a convertible note that will convert into the upcoming Series B. And it will basically fund us for the next year as we're growing our commercial side. We're hiring salespeople. We've proven the model on a small number of salespeople. So we're going to double to the big number of 4, the number of salespeople that we have, because we actually have more leads than we can possibly follow up with. We are also in the process of acquiring a small profitable company that is in our field. It's a leader in environmental controls in cell culture, and environmental controls is one of the most important pillars. And we expect to raise our Series B March, April, May of next year for 15 million. That is the last round that we expect. Most of our efforts now are on software. We have very tightly integrated hardware and software, but we've developed this platform and actually have developed several products. Next slide. So our products are instruments and software to provide automated imaging of cells, tissues, stem cells, so that we have reproducible data, comparable data. And I want to read what one of our customers who is using our instrument for COVID-19 and other infectious disease research. So he's talking about the current state of the industry. Current tools are failing us because they hurt the cells, only give us 2D, single point in time data, and don't capture contextual data or enough data. In fact, when an experiment goes wrong now in the lab, we don't know why. There's not data to trace what happened, not documentation to go back and look, and documentation combined with images of the cells. After using our instrument, he said, Thrive will change the world. Thrive allows us to understand and see living dynamic cells and tissues in 3D over time with remarkable images. Next slide, please. So let me tell you just briefly about the state of the field of cell culture. So cells are kept in these incubators and they're just stacked, and somebody goes in and pulls them out, disrupts other people's cells, crosses multiple environments, takes them to a microscope, spends several hours, on average, researchers spend several hours a day looking at millions of cells. And then they take the cells and put them back in the incubator. And guess what? No data is retained. It's only what they remember or might scribble in their notebook, or they might use a digital microscope to take a few images, not the thousands of images that we take, the gigabytes of images that we take in minutes, and the same way every time. The field is in such disarray because it's been manual for 70 years that mislabeling rates are actually up to 30%. And those are FDA and NIH numbers. That means 30% of the time, researchers are studying what they think might be liver cells, but are in fact pancreatic cells. So no wonder we have such high rates. And it sounds extraordinary and nobody thinks it's in their lab, but it's the accumulation over 70 years of a little bit of mislabeling, here and there. So without our instruments, you're just unable to reliably conduct these experiments. Next slide, please. So what is the result of this? It's called the reproducibility crisis. And it's not that well known outside of science, but Nature magazine did a survey of 1,500 scientists, and 52% said that there is a significant crisis. The title of that was 1,500 Scientists Lift the Lid on Reproducibility, the Crisis Rocking Research. Imagine a field where half of the people don't believe the results that are generated in their field. I started this company because of an article I read in 2013. Amgen did a study with 100 scientists over 10 years to try and reproduce 53 studies, landmark oncology studies, and they were able to reproduce 6. So 89% were not reproducible. Bayer had a similar, a little bit better, 78% were not reproducible. No study has found that less than half is not reproducible. That means half of our 50 or so billion dollars that we spend in the US in preclinical research is really misguided. And another study showed that about half of that is linked to cell culture. That's where all of these experiments start. And often, unfortunately, the bad science isn't discovered until it's in a clinical trial, and a lot of money's been spent. So this is one of the major causes that slows down drug discovery, drug development. It also puts patients at risk because hundreds of thousands of patients have been enrolled in trials where, in fact, the science was not reproducible. Next slide, please. So I want to give you a couple of examples of where we have solved the reproducibility problem. So you all know Goldfinger, a martini, shaken, not stirred. Well, I have the boring life science sequel for that. Do you like your cells shaken or stirred? So this is really an amazing situation. Dana-Farber and University of California, Berkeley, 2 labs were cooperating on a joint NIH grant, and were not able to duplicate each other's data, could not reproduce each other's data. It took them 2 years to figure out why that was. And it turned out that one was shaking their cells and one was rocking their cells. Little things are important. There's the details, there's a devil in the details. And another example, next slide. I mentioned to you the mislabeling of in cell culture. Well, one of the leading research institutes in the world, I won't name their name, by looking at their data generated on our instrument, was able to see that there were some cells that were growing at a faster pace. They were outliers. And so they went back and looked, and sure enough, they were mislabeled cells. So having data, next slide, please, and even a simple documentation system like we provide, which is, we brought barcodes to the cell culture lab, which amazingly has never existed in these labs. There's very little there. The bar is extremely low. But just by putting barcodes on the instruments that are used, the reagents, the plates, the operator of the lab notebook, solves those problems in those labs. One of our customers points out that it really saves them time and money. They don't scramble around trying to figure out who did what. Next slide, please. So these are the products that we're going to solve this problem with. On the left are the 2 products that we have on the market, a benchtop imager and a standalone automated imager with 50 plates. Both of them have best in practice software. They're networkable. You can, from your app, control these instruments, and they're generating for the first time really massive, consistent, comparable sets of data and documentation, which just hasn't been available before. Most of our efforts now are on a variety of software modules where we're focusing on the most important workflows, such as infectious disease research, plaque assays, imaging organoids. And the architecture for all of our products is the same. The imaging has been built, the databases have been built. For our customers, any of these instruments are building increased value because they can go back and look at what happened. Next slide, please. So there are a couple of things that we do differently than the rest of the industry. So number one, we're offering a full solution. Right now, it's basically the microscope. But to solve this problem, you need to solve a number of problems, and I call them pillars, and that's what we're doing. So you need to be able to control the environment. You need to be able to manage the projects and document it. And we provide tools that allow researchers to be much more efficient and to automatically document and set up their experiments. Obviously, acquiring images is important, and we do that better than anyone. We take images at 100 focal planes, literally 50 gigabytes of data in 10, 12 minutes each time that it scans a plate, and it does it the same way every time, so that you can compare your images over time. You can compare these images with another researcher. So all of this is automated, and we put it all in a database, a huge database. Terabytes are being generated that's referenceable and accessible. Imaging, next slide please, is really the crux of the matter. We're the only instrument that can find and track a single cell over time or a single colony. It was a holy grail. After CRISPR, which is gene editing or single cell sorting, you're not able to find that cell until it's grown enough that it's a big enough clump. And so we know nothing for a couple of weeks about whether even the experiment worked. We take 100 focal planes at least, so we're able to image up to 4 millimeter tissues and organoids, which no one else can do. It also means we can image things like organ on a chip, also suspension cells, which is the basis of most immunology. Basically, we see things that have never been able to be seen before, as one of our customers put it. We're producing genomics quantities of data, but for live cell biology, which has never been done before. Next slide, please. So we don't have, actually might be slide. Nope, sorry, you're right, Mark. Competition is basically the digital microscope. And what it lacks is the ability to take these stacks, to control the environment, to fit into the workflow, to do it automatically so that instead of spending, I mentioned to you, it's actually 2.5 hours on average per day spent at a lab looking through the microscope. It's done remotely. You put the plates in or you have the plates automatically put in. And we have a documentation system. Next slide. So we don't have the FDA as a barrier, which is actually a useful barrier. So we focused on, besides having great products and happy customers, which is one of the best barriers to entry, really enormous IP portfolio with 86 patent applications, 28 of which have been issued. And we cover the whole field. We cover it for research, for manufacturing, even, for instance, a big area is in vitro fertilization. Those are cells that people really care about. And so we also cover software, image processing, blockchain, which is actually has a useful application to integrity of data, Internet of Things, because we're really tying together everything that's going on in the lab. Next slide. So we offer benefits to both the users who are culturing, the cell culturists, as well as the researcher. And one of our customers pointed out, we have a complete lack of tools to help us in this increasingly important task of cell culture. We help produce better cells. We like to say good science needs good cells. But we also, in taking all these images, allow researchers to have breakthrough insights. And in the future, there'll be a third use of this data, IA, which is industrial automation instead of AI, which we use. These databases are going to be used by us with increasing amounts of robotics and analytics, and to drive the most intelligent robotics that are available. There are some robotics that are available, but they're mostly programmed and take a full-time programmer. Next slide, please. So our customers, I mentioned, are about 60% are pharma. This is includes orders. And the most common application is drug screening and regenerative medicine, particularly stem cells, which are very, very difficult to grow. Next slide. You know, Marty, we discovered through customers. And it's, I mean, when the microscope was first built, I think it was, basically for following amoebas. And look at what it's used now. We had no, we thought we had a lot of good ideas, but it was actually the Broad was able to screen 10 patient-derived cell lines with 30 different drugs, collect that data. They've never been able to do that before. So here are some examples of images and the fields that we focus on. So organoids, drug screening, the viral plaque assay, characterizing stem cells. Next slide. Just want to show you a couple of images. So these are organoids, which others can't image. And this is the fastest growing area of cell cultures. So rather than studying drugs, cells on the bottom of a plate, we study them in organoids that are closer to tissues. The next slide, please. So this is the first paper that we've had, and this is a drug screening application that never could have been done previously. So basically, by automating and following the repair of cells in something called the scratch assay, the researcher was able to screen over 24 drugs. They were never able to do this because they would look to see how the wound was being filled, and they were doing it manually, and each person had a different count. Even the same person looking at the same image at a different point in time would count it. So to have these kinds of metrics just haven't been available. Next slide. And actually the next one. Thanks. So this is the viral plaque assay, and this was done by the person who said Thrive will change the world. We will completely change the way infectious disease research is done. The viral plaque assay right now puts a layer of viruses on top of cells and counts the holes that it produces. And at one point in time, by staining the cells, which kills them, we're able to not only see them before staining, so sooner, but actually track the progress. And there's a huge amount of data there that's never been available before. Next slide. So this is an example of tracking a single cell from the Harvard Stem Cell Institute, and you can see it growing from a single cell into a colony. The next slide, please. So I didn't have a chance to mention my co-founder, Alan Blanchard. So Alan worked for 25 years in genomics, developed most of the instruments that are used in the field. And basically, we met at a cell culture lab that a friend introduced us to, and we were horrified because it hadn't changed from when we knew it decades ago. And after 25 years in genomics, he decided, let's bring the same kind of data to cell biology that was brought to genomics by sequencing. And that's a several multi-billion dollar companies, and we expect to follow that same path for live cell biology. Next slide, please. So I mentioned the offering. Sal Buscemi, who I met here and is speaking, and Albert Yuen at HRN are leading a round of family offices. We're going to raise another $3 million. The discount will be approximately 30% into the upcoming Series B. And please talk to Sal. I think you're speaking later. And we'd love to have you join that. The next slide. We fit beautifully into 25 or so companies as potential acquisitions. We've been contacted by almost all of them, some several times. We fit into life science tools, research, microscope companies, instrumentation, drug companies, CROs, or contract research organizations. Next slide. So in summary, we have the solution to these problems. They don't require discoveries, and we're planning to transform this field and have 90% market share like I did at Cytek. And ultimately what we're trying to do here is bring medicines from the lab to patients more rapidly. I think we have the team to do it. I rather look at what customers say. So we hear all the time things like remarkable resolution. This is what cell culture and cell imaging should look like. So my contact information is here, tom@thrivebio.com. Would love to have your assistance in raising funds, in finding customers and collaborators. Thank you, Marty, for having me again. Sure. So what is a distribution strategy and exit strategy? So we're selling into laboratories which are used to purchasing fairly complex equipment. Our sale prices on our instruments are 100,000 to 250,000. The software modules are about $35,000 each, but it really requires a direct sales force. However, we are leveraging it with, there are some nationwide distributors in the US that have additional kind of feet on the street. And internationally, we're going to use distributors, initially. I think it's probably 2 or 3 years until there's an exit. Who knows? It could be sooner, it could be later. I think that the value in Flatten really begins to increase dramatically in 2 or 3 years, by which time we'll have 250 instruments, a lot of research papers that have been used, proven use cases, which we actually already have, of where we're saving time and money. And most companies in life science tools, although I've run public companies, I'd love to go public, the majority get acquired by others. So Illumina, the leader in sequencing, in genomics, actually acquired their instrumentation, and my co-founder was involved in the development prior to Illumina. So that it typically is a multiple of revenue. So for instance, Cytic was 10 times trailing revenue. So we sold my company for $6 billion after taking it public. That was FDA regulated. The non-FDA regulated ones are at a higher multiples. It's actually, life science tools is one of the best return area. It's also the smallest and is ignored by a lot, in part because people they can identify with a drug that's being developed to cure something that was somebody's family member struggled with. In fact, we help all of those companies. We help drug discovery in all the fields to be done better and faster. Sure. So we don't have a set pre or post money valuation for the Series B. The current round is at a discount to the upcoming Series B. We do, however, have a term sheet that is at about $80 million pre-money. We're probably not going to take that because we didn't love all the terms, but it was close. Yeah, so that's assuming. Yeah, so that's assuming no sales, which we know that we'll have. So that's really, I should have mentioned, that's really kind of a worst case sort of scenario for us. We have really fantastic investors. Most of them were my bosses or previous investors, myself, who will back the company. We have several families who've said, when you raise the next round, make sure it's the right people. If we don't like them, we'll take the investment ourselves. It's important to have the right group. Family offices are really just phenomenal. They typically have been in business, understand business, even if they're from a field outside of life science. Really wonderful to have.